Hey everyone, last time I looked at a recent paper on HARs, human accelerated regions. Evolutionists say that these regions of DNA evolved rapidly when evolution was constructing the human lineage. We saw that there was a problem with this, a problem for evolutionary theory caused by these HARs regions. The problem was there's just too many coordinated mutations required to construct the human lineage. These HARs show that. This time I want to show you some actual HARs sequences. Have a look at these and you'll see what I'm talking about. And also show you another major problem that HARs presents for evolutionary theory. So let's have a look. This is a paper from 2006 that shows some example HARs sequences. So let's have a look. Here's one example HARs. Before we look at this, let me remind you our DNA is made up of 4 nucleotides: adenine, thymine, guanine, and cytosine, and this graphic illustrates those. We abbreviate these 4 nucleotides A, T, G, and C, and so that's what you'll see when you see DNA sequences in a journal paper. You'll see the A, T, G, and C abbreviations, and that's what we see right here. You see these sequences. What we're looking at is a HARs sequence from the human, chimp, gorilla, and so forth genomes. So let's zoom in here, and you can see those abbreviations, the A, T, G, and C. What you'll notice is that in some cases, in these bold cases, the nucleotide is the same. It is conserved across these primates, but it's different in the human. So here you've got thymine, 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 and then in the human you have a guanine. And so that happens a few times in these HARs sequences, and evolutionists look at this and conclude, oh, this evolved rapidly because it was the same until you got to the human lineage, and then it evolved. It changed. The nucleotide changed. And evolutionists conclude that it evolved rapidly because there's a relatively short amount of time between the gorilla and chimp and then to the human. But this is it. There's no other evidence to conclude that there was some evolutionary event. It's simply a difference in the nucleotide. You have a guanine versus a thymine in these other species. This is the only evidence evolutionists have, and they conclude, oh, there was rapid evolution. I wanted to show you this so you can see how paltry the evidence is. It's simply a different nucleotide. You have thymines in the primates and then a guanine in the human. There's no evidence that there was any evolution going on. In fact, there's an enormous problem with that idea. But evolutionists conclude, oh, it must have evolved. There's a different nucleotide. This is evidence for rapid evolution. There's no evidence for this except for the fact that there's a guanine in the human sequence here. So I wanted to show you that. Here's another example of another HAR. About 10% of this sequence has been changed in the human sequence, in the human DNA sequence. Okay, so that gives you an idea of what these HARs look like, but it gets worse here. And let's go back to our paper here that we looked at, that came out recently, looked at last time. I want to show you this one passage here where they explain that what exactly are these HARs regions. Now, in the DNA, some of the regions code for proteins, and they're a small fraction, and those are called coding regions. And then you have the remainder of the DNA, which is referred to as non-coding regions, and they do a variety of things in the non-coding regions. So here it's explaining that HARs are mostly non-coding and are enriched for transcription factor binding motifs, suggesting that many HARs potentially function as human-specific regulatory elements that coordinate precise spatio-temporal patterns of gene expression. Now, if that was confusing, let me give you the boiled-down version. So HARs are non-coding. They don't code for proteins. One of the major functions of the non-coding regions is regulation of the coding regions. When do you turn a gene on? When do you turn it off? If it is on, maybe it needs to be paused, and so forth. Regulation is very complicated. There's a lot we have yet to learn about it. There are many gradations and variations on it, a lot of different ways that the cell uses to regulate genes. Many of the HARs serve in that regulatory role. Okay, so that's interesting. So, in other words, to get to the human brain, you had all these HARs regions undergoing rapid evolution, and they were serving a regulatory role. In other words, the proteins were already there. All of the proteins were already in place. You didn't need new proteins, you didn't need new parts, so to speak, to create the human brain, to get to the human brain from the other primates. What you needed was regulation. When should I use these proteins? Where should I use these proteins? So the parts were there, we just need to learn how to use them, in other words. That's a lot of serendipity. Let me give you an example here. Imagine you came across an old shipwreck, you salvaged the parts, and from those parts you made a new fancy new car. That's kind of the idea here, that the parts are already there in this ancient DNA, and you just needed to learn how to turn them on at the right place, and so forth. You needed to regulate them in order to come up with a human brain. There's a lot of serendipity there that's required to believe in evolution. Religion drives science, and it matters.